I want you to imagine your DNA is a shoelace. The three-dimensional folding of your DNA is not random, and neither is the way you tie your shoe. Your DNA is folded and packaged in a highly organised manner into several layers, and this allows regulatory regions in the DNA and genes, normally located quite far apart, to be able to interact. This is extremely important for a cell because it allows it to function properly. Similar to when your shoelace is tied properly, it means when you walk, you won't trip over. In my PhD, my research aims to understand this three-dimensional DNA network because it is highly disrupted in cancer, and that prevents a cell from functioning properly. Just as when your shoelaces are not tied properly, it means you'll trip over. So to investigate this DNA network, I am focusing on a protein complex called SNF. The role of this complex is to modify the packaging of DNA at its smallest level, and it is highly mutated in many cancers. Now, I've hypothesized that DNA packaging at this smallest level will also impact the overarching final three-dimensional DNA network. And that is, how does the first loop you make with the shoelace affect the way you tie the knot? I'm first analysing this relationship in normal cells, then we'll look to see how it is altered in cancer because of the mutated SNF. I'm also investigating epigenetic drugs that are currently in clinical trials for cancer, and I hope to use these drugs to reverse any abnormal DNA packaging because of the mutated SNF and return the overarching three-dimensional structure back into its correctly folded network, similar to retying that shoelace. My research is critical for understanding the mechanisms behind the misfolding of DNA and will repurpose current cancer therapies to target the abnormal SNF and treat a greater range of cancers so that I can keep that shoe tied on your foot and stop you tripping over.